recording? No, not yet. Oh, I am. Okay, good.
All right, okay. I'm pretty sure the stuff I put up is going up properly to YouTube and stuff. Has anybody looked at these things? Okay, you can wait till the exams. They just start looking at, oh, what the hell is this thing? Okay, by the way, what we're going to look at again, we're looking at regulation of transcription in eukaryotes. Now, we mentioned on Tuesday, we gave a sort of generic, and we'll do this again later on, a generic eukaryotic gene showing the key G sequences involved in the process of transcription. Okay, so we're going to do that. Hang this, Wallace. Don't say I never gave you anything. I can find it. Ah, here it is. Okay, so she faced the cold and the snow. Neither cold nor snow nor dark of night will keep you from doing class. Okay, that's good. I like that. It's not like 101. Nothing will keep them coming into class. Okay. Yeah, all right. Anyway, we looked at some of these key sequences that were involved in regulating and activating transcription, and we'll do that again later on. But what we want to look at now is, in eukaryotes, you're not dealing with naked DNA. It's constantly complex with proteins that make what is called chromatin. Now, of course, the chromatin inside an interphase nucleus. Of course, our DNA is organized in the form of chromosomes. Chromosomes are these linear pieces of DNA. In other words, they have open ends. They're not circular like you see in prokaryotes. And each organism has a number of different chromosomes depending on time. Some have as little as 2 or 4. Some have as many as 200 separate chromosomes. We've got in, of course, diploid cells, we've got 46 chromosomes, 23 pairs, including the X and Y, which are quite a bit different from each other. Okay, now, in the interphase nucleus, the chromatin is spread apart. So you see a mottled appearance in the nucleus. We remember this from our slides the other day. During mitosis and meiosis, the chromatin undergoes a reorganization and they compact to form these mitotic or meiotic chromosomes. It's the same material, just packed in differently, so now it picks up the stains. You can see them very clearly and visibly. In the interphase, they're splayed out all over the place, and they're very, very hard to trace. Okay, now what is chromatin? Of course, chromatin compacts, makes the chromosomes. But chromatin has a basic structure. And there's a whole bunch of other proteins that are involved in regulating and modifying that structure. We get chromosome condensation. And not only does chromatin have this structure that organizes the chromatin, organizes the DNA so it doesn't get tangled up and things like that inside the nucleus, but we're also seeing that chromatin, especially the major proteins involved in it, play a very important role in regulating transcription. And we're going to see this in just a minute. So let's take a look at the basic structure of chromatin. There's several different layers, a hierarchy of structures. There's several different layers of structure. But we start with the simplest thing, and this is what's called a nucleosome. The nucleosomes are the foundation structure of chromatin. Now what it consists of, you look at a nucleosome, what you see is sort of a lozenge-shaped complex of proteins with DNA wrapped at 1 3/4 times around it. Here's the DNA. And that turns out that's 146 base pairs that associate with this. Let me get this to you, Mr. Prince, if we can find. Ah, here it is. Okay, Jeremy. Here we go. Okay. And then you have a spacer that's variable. It goes from about 20 to about 80 base pairs of DNA. And then you wrap around a second one of these. And you continue this process on all down the way. These DNA protein complexes are the nucleosomes. And this is our spacer DNA. In other words, it's not associated with the nucleosome. Oops. Now the 146 base pairs wrapped around each nucleosome, that's pretty consistent. That's pretty universal. The spacer DNA varies from organism to organism, cell to cell, even, one part of the DNA and another. Yeah?
What's the middle part as what DNA?
Spacer. In other words, it's a stretch of DNA that's not associated with the nucleosome. Okay, now the question is, what makes up the protein component of the nucleosomes? Well, the key thing is a series of proteins known as the histones. Histones are very basic proteins, meaning they're loaded up with positive charged amino acids. They're extremely rich in positively charged amino acids. And of course, if your idea is to stick with negatively charged DNA, that's exactly what you want. So they're highly basic. They're small. They tend to have molecular weights of about 15 or 20,000 or so. They're fairly small. And an important thing about them is the histone proteins are highly conserved. Now what that means is the sequence of amino acids varies very, very little from one organism to the next. In fact, if you take something as divergent as, say, protohistones and human histones, you only find 1 or 2 amino acid substitutions, even though you're talking about a divergence time of, 700–800 million years. So, they're highly conserved. Now, most proteins, of course, through evolution, you get all kinds of variations and stuff. So, for instance, the sequence of the globin proteins that make up hemoglobin in humans is going to be noticeably different from that in mice or a frog or a fish or something like that. Now, when you have a highly conserved protein, it doesn't mean that there's not much mutations going on in the genes that code for that. What it means is that the protein must be so fine-tuned that even slight changes to its amino acid sequence will cause it to simply not work properly. In other words, it's intolerant to mutation. You get a mutation in histone genes, chances are that it's going to be lethal. Okay, so very highly conserved proteins. Now, an interesting thing when you look at the histone genes, we mentioned before that the vast majority of eukaryotic genes, the genes are separated into coding regions called exons and non-coding regions called introns, kind of like the show and the commercials. Okay. Histone genes are some of the few eukaryotic genes that do not have any exons, or rather any introns. It's a continuous coding sequence. And the idea behind that is it's just too much of a risk to take a chance that you're going to have an accident splicing the exons together and come up with something different. It's just too much of a risk, so let's not have any introns in those genes whatsoever because we don't want to take chances. You switch a single amino acid in a histone, and chances are you're dead. Okay, so those are a couple features of the histones. They're very highly conserved proteins. They're very positively charged, and they're all relatively small. Now, the histones assemble together to make up this core particle of the nucleosome, and the DNA wraps around it. So, turns out we have 4 histones that go into it. One called H2A, H2B, H3, and H4. And there's 2 each in each one of these nucleosomes. They kind of form half nucleosomes. One of each forms sort of a half nucleosome, and the 2 halves assembled together, you got the full nucleosome. Okay, so that's the basic structure of chromatin, are these nucleosomes. Now, without going into too much detail yet, the nucleosomes the DNA is bound so tightly to the nucleosomes that if there's a critical sequence, say a promoter over here, okay, let's suppose this is a promoter. If it's wrapped around a nucleosome, transcription factors in the basal complex and the RNA polymerase can't get to it. So, this promoter is useless for transcription. Nothing can stick to it. It's already bound tightly to the nucleus, tightly to the histone core. So that says something. That says that these nucleosomes are inhibitors of transcription. Remember, we said in most cases in eukaryotes, genes have to be activated. The default is that the gene is not being transcribed. And if you want it transcribed, then you have to do something to it to get it transcribed. Okay, so in this case here, the nucleosome is going to prevent transcription because the key sequences are buried and wrapped up around the histones. Okay, so that's an important thing. And it also tells us that if we want transcription to occur, we're going to have to do something to those nucleosomes. Either move them out of the way, get rid of them, or what have you, so that the sequences like promoters and stuff are accessible to the appropriate proteins. So that's one thing. Now this is sort of our foundation level of chromatin structure, the nucleosome. And we'll go back to that a little bit later. But we can also assemble things into additional structures. If you take a look at nucleosomes in an electron microscope, you see this thing, and it looks kind of, it's often called the beads on a string configuration. It's a shrunken down version of what I just drew in a lot of enlargements. Okay, so this is okay, the beads, of course, are actually the nucleosomes, and the string is the DNA. So what you're seeing is the nucleosomes and the spacer DNA going from one to the other. Okay, now, there are a couple other kinds of histones that play a major role in joining the nucleosomes into more levels of structure. And these two include a family called the H1 histones, and their close relatives, the H5 histones. H5s are kind of very similar and can be viewed as a variant of the H1 histones. Now, those are not part of the core nucleosomes that we saw. But what they do is they will link one nucleosome to the next and kind of stick it together, glue it together. So if we were to do something here, I'm going to make a few nucleosomes here. Okay, here's some DNA wrapping around, a little bit of spacer, and it wraps around again. Now what we can do is things like H1 or H5 will link between the two nucleosomes, and under certain conditions it can pull them together, and then the nucleosomes get packed more tightly. So if we add some H1,
Was it getting rid of the spacer?
well, it kind of pulls it together. Spacer is still there, but it's just packed in more tightly. So now what you do is you get something where the nucleosomes are kind of arranged, stacked up on top of each other, kind of like so. And they often call this the 10 nanometer fiber. It's the nucleosomes packed together. And the H1 and H5 tend to play the major role in that. So we have this 10 nanometer fiber. Okay, now in other chromatin proteins, and H1, H5 play roles in this too, that fiber can then form a larger fiber called the 30 nanometer fiber. Now exactly how that's organized is a subject of some dispute. At one time, people thought that the 10 nanometer fiber kind of wrapped around to form a spiral coil that was hollow in between, what they call a solenoid. Anybody know what a solenoid is? It's you take like a hollow tube and wrap some wire around, and then you run electricity through it to get magnetic field pulls things into the tube. That's what a solenoid is. It's commonly used in a lot of electronics. Okay, that's what they thought at first, that this 10 nanometer fiber made a spiral coil, so you made this solenoid-like structure with a hollow core. But there's a lot of dispute on that. I think what's probably leaning in is actually that fiber kind of does a zigzag back and forth to form our 30 nanometer fiber. So now you're going to draw it like so, zigzag back and forth. And this may be more accurate representation of that 30 nanometer fiber. You're kind of bending the 10 nanometer fiber back and forth over and over and over again to make that 30 nanometer fiber. Now if you take it, yes?
So do the H1 and H5, do they join the 10 nanometer together to make?
Probably, but we don't know the molecular details.
Okay.
Okay, and part of it, we still don't know. You'll find partisans for the solenoid structure. You'll find partisans for the zigzag structure. The stuff I've read, I would support the zigzag structure myself. But, you know, you still have people that like the solenoids. I think the evidence is more in favor of the zigzags. But, so we really don't know exactly how it's held together. We just don't have that level of structural detail yet. Now, if you took, for instance, if you took a mitotic chromosome and squashed it out, and looked at it under an electron microscope, what you'd see is it would look like a skein of yarn, and the yarn would be the 30 nanometer fiber. So that's clearly a very important structure of chromatin. Now the question is, how do we go from this to what we would consider a chromosome? And that's where you have more speculation than evidence. What we see is that each chromosome has at the center of it a core of a number of proteins, especially something called topoisomerase II, which actually also involves in untwisting DNA during certain things. But there's other proteins in there as well. So we have this protein, what's called a chromosome scaffold. And it's a protein. Now one kind of, I guess, as close to a consensus model as you can get on this kind of stuff of what goes on, is that the 30 nanometer fiber attaches at certain points to that chromosome scaffold and then forms these large loops. Now this here is our 30 nanometer fiber. Loops out, connects again, loops out again, and so on. Okay? Okay, so that's a 30 nanometer fiber. And they see that these attachment points tend to be about a few tens of kilobases. In other words, a few tens of thousands of bases apart. So that might be the next level. Now, it's possible that you have, and this is almost going to be impossible to draw, but let me try it anyway. What the hell, right? Okay, here we have one loop. And then in this, we can picture this in 3 dimensions. We got another loop here, another loop here, and it kind of forms like a sort of spiral rosette and then continues on and so on. That's kind of as close to a consensus model of a chromosome as we can get. Of course, it's all kind of tangled up in there anyway, so it's really hard to determine exactly how it's organized. There's good evidence for that 30 nanometer fiber being attached to the chromosome protein scaffold several tens of kilobases apart for each attachment point. Exactly how the rest of that organizes is still a matter of dispute. But that's one kind of model. They just have these spiral coils of these loops of that 30 nanometer fiber and just spirals around the chromosome scaffold, and bang, lo and behold, you got finally a mitotic chromosome. That's kind of a reasonably good model for that type of stuff. Okay. All right, so that's one thing we see for basic chromatin structure. Now, we might want to ask ourselves, we see that these nucleosomes often bind to and block key sequences involved in transcription, thereby inhibiting transcription. So that suggests that we have to do something to the chromosome, to the chromatin structure, especially those nucleosomes, before transcription can take place. Because otherwise all the goodies, the proteins that we need for transcription to occur just can't get to those sequences. So that's the next type of thing.